Matthew Niemiller from the Department of Biological Sciences of the University of Huntsville of Alabama. Um, Matt is a colleague and a friend of mine. We go way back. I guess you were a graduate student, I think, when I interviewed for a job at Middle Tennessee State University,
Yeah, like circa 2005, I think.
2005. Yeah,
Yeah.
so we're talking 20 years ago now. Um, Matt's a salamander colleague of mine mostly, but he's done some incredible work. He started working on cave salamanders to begin with. There's an incredible world of biodiversity that Matt's been working on basically for his entire career. It's kind of started as kind of a side project, has led to basically a huge career for you. And so today, Matt's going to talk to you about some of that work at least. He'll be talking about diversity in the darkness and addressing our gaps in the knowledge of subterranean biodiversity in North America. So please feel free to stay afterwards. I'm sure Matt will be happy to field questions, and I'll try to keep a look on the chat here if any of you have questions during Matt's talk. So without further ado, take it away, Matt.
Thanks, Ken. Yeah, thanks everyone, and thank you for the invitation. Yeah, just for the students and might be grad students now. Yeah, my first master's project was actually on tiger salamanders of all things. I was supposed to radio track them at Arnold Air Force Base in Middle Tennessee. After a year of field work and spent, gosh, hundreds of hours actually in the field that year, we caught six salamanders. They were all too small to put transmitters in, so I was basically without a data set and looking for kind of a new project. And just by sheer chance, my master's advisor, Brian Miller at Middle Tennessee State, had a state wildlife grant with the state of Tennessee to reassess conservation status of the Tennessee cave salamanders, Tennessee state amphibian. And that's honestly how I got started on this path. And little did I know, over 20 years ago that I'd be, still working on these systems. And I just kind of like fell in love with the amazing diversity and really our lack of knowledge. We really just had, that's what I kind of want to focus on today, talking about is we know a lot, but there's a whole lot more that we still don't know, and we need more folks working on this amazing diversity that's literally out of sight, out of mind because it's underground. So a little bit of background, right? So the systems that I work in are in karst terrain, right, which is a special type of landscape. They're at the surface that is largely formed through soluble rocks that can dissolve, right, through weak, carbonic acids. So it includes things like dolomites and limestones and marbles and whatnot. And this type of terrain, though, is actually quite common on the Earth's surface. It actually makes up about a fifth of the Earth's surface worldwide. And much of the world's drinking water actually comes from this terrain. So roughly about a quarter of that. And predictions suggest that if we continue to see current changes in climate, that our reliance on basically groundwater is just going to increase, over the next few decades. Within the U.S., what I largely work in are the larger cavities, right? The cave systems. There's at least 70,000. That number is probably well, more than that. Most of my career has been working in Tennessee, where we have over 12,000, almost 12,000 caves now. So Minnesota's right, still the land of 10,000 lakes. Well, Tennessee's the land of, 12,000 caves. So to give you an idea where this karst terrain and where these cave systems potentially develop at, this is a karst map of the United States developed by Dan Doctor and colleagues of the USGS several years ago, and this kind of highlights. I want to highlight there the areas in blue. Right, this is the carbonate, basically karst. So these are the karst areas. Where they're largely found closer to the surface. Obviously, there's a lot of different types of pseudokarst and evaporate karst, and other volcanic karst out west. But some areas stick out, right, on this map. You see Missouri, much of this southern part of Missouri extending into northern Arkansas and Oklahoma. They're the Ozarks. You've got the interior low plateau, essentially from southern Indiana down into my neck of the woods in Alabama. You have the Valley and Ridge. Florida, basically, it's all karst. You might not necessarily recognize that, but even out by you all, right? You guys have karst in Minnesota, part of the driftless area. So that kind of expansive karst there that remained largely ice free during the Pleistocene. Right. So, there are some cave systems in Minnesota, associated with Wisconsin, Iowa, and extreme, northwestern Illinois. And these are beautiful landscapes, right? Beautiful terrains. These are some, photographs by a good friend and colleague, Chuck Sutherland, largely from Tennessee. but they're just, amazing environments. And hopefully some of you have been able to go into a cave before. But it's not just caves. We have, karst springs, seepage springs, a lot of other types of subterranean habitats in which this life ultimately kind of resides in. Some really, really beautiful areas is Stevens Gap in northern Alabama, real popular area for like wedding photography. So you see that kind of waterfall coming in, the light coming in. There's a little pillar there where people like to stand and get their photos taken out. And we like to explore underground. It's one of my grad students doing, Superman pose. I mean, they're beautiful landscapes when you really start to look at the geology, right? Just the amazing rock formations. In general, got stalactites, stalagmites, columns. A variety. We could spend an entire semester just talking about, right, the geology of caves and life underground. This is an actual photograph of me doing work underground. My students don't believe it sometimes. I spend way too much time behind a desk with report writing and grant writing anymore, but I do occasionally still get underground. This is during a cavefish survey at a cave here in Alabama. What I want to kind of start out with, though, is talking about, well, why should we even care about, these habitats? Why should we care about, cave and karst? And that really, gets down at its core, right, to the ecosystem services, those basically services that ecosystems and, biodiversity can provide us freely to human society, right? And so those can be broken into, right, things like provisioning services, so the raw materials and foods and whatnot. So I'll kind of talk about a few of these as well, the cultural aspects, right, and the regulating aspects. So first off, you have groundwater. I mean, these are major, in many cases, major underground aquifers in these karst terrains, which we rely on for drinking water, for our agriculture, watering our fields or our cattle and whatnot. And again, as things potentially progress with climate change, we'll be even more reliant, on this resource. Historically, a lot of mineral resources occur in these karst terrains, and that includes, mining of dolomite, marbles, limestone, things like gypsum. In some parts of the world, still guano, bat guano is being mined, from cave systems. And then we think about the historical kind of aspect of this. Saltpeter during the Civil War area for gunpowder was being, mined from cave systems actively. We have a number of important archaeological and cultural resources there. Many cave systems throughout particularly the southeastern U.S. were utilized by Native Americans to varying degrees. In some cases, they're really, spiritual sites and of great importance, in general. This is kind of showing a span for like Civil War era there in the left. Got a pictograph that probably dates back about 900 to 1,000 years there in the middle, and then some more modern art, probably circa 1997 in a cave system, from spray paint graffiti. So we see this in some cases, right, repeated use by humans in some of these cave systems that dates back even a couple thousand years. Of course, many cave systems can be important sites for paleontological resources. So it includes, fossils that are dating back, anywhere from, 150 to 400 million years old in some of these layers. But the same token, we see a lot of Pleistocene fauna that's being recovered. Even Pliocene fauna is being recovered from some of these cave systems. So caves, the pits, they act as they're basically natural pitfall traps. Things fall in, they can't get out, and with the right conditions, right, you see a lot of preservation of these remains. And so that's allowed us to, make inferences on past communities in many, many areas in the Southeast and of course across the U.S. in general. There's also the recreational aspects, particularly in my neck of the woods. We have a lot of really deep pits that cavers love to explore, anywhere ranging from 100 to almost 450 feet. Um, Folks, love to go caving. It's big business, big industry, down here. A lot of really, really great sites. Some of it's for, looking at those formations. Some of it's for the sport of exploration. But for me, it's always been about the biology. So, that includes things like the microbes, right? The bats, but the kind of that group there in the far left is what I spent most of my career trying to studying, and it's the cave obligate fauna. So these are the species that are found just underground and really nowhere else, and they have unique adaptations that we call kind of trogomorphy. That includes morphological features like tendencies to have reduced pigment, reduced or loss of eyes, changes in their physiology. They tend to have longer lifespans. They tend to be more starvation resistant because these resources are these environments, right? They lack light, they lack primary productivity in the form of photosynthesis, and they're fed almost entirely by energy from the surface. And so, because of that, energy can be quite limited in some of these systems. But there's a remarkable diversity that we're still discovering, and documenting that lives in these habitats. And they provide, again, a number of important, services. So just some of the regulating services that have been, suggested, typically with respect to kind of groundwater fauna, important in nutrient recycling. They can be great indicators of overall ecosystem health, right? Particularly, drinking water. Some of these species appear to be highly sensitive to any sort of perturbations to their habitat. And so if something's happening to them, it's kind of the canary in the coal mine, right? It's kind of a red flag that, hey, something's going on here. And, when we have, and particularly in rural communities, humans, relying and people relying on this drink for drinking water, at springs or wells, something we need to be paying attention to. And of course, our bat populations, they provide billions of dollars in ecosystem services with respect to pest control, and importance in some areas with respect to pollination and seed dispersal, particularly out west. But it's an amazing, amazing diversity. This is just some of the terrestrial species that we consider kind of cave obligates. Most of these species are arthropods, so they're invertebrates. So things like millipedes and a variety of arachnids, from mites to spiders to pseudoscorpions to harvestmen. With respect to some of the insects, we have a great diversity of beetles. We have diplurans, another hexapod group, springtails, terrestrial isopods, so basically terrestrial roly-polies, and then things like snails. With aquatic fauna, it's dominated by crustaceans, so particularly isopods and amphipods. We have ostracods, copepods, decapods, so the shrimp and crayfish. We have flatworms, aquatic snails. And in terms of vertebrates, we actually don't have that many vertebrate species that we consider kind of cave obligates, but there are several fish species, about a dozen of those, and about a dozen salamander species. So what's pictured here is a southern cavefish and Tennessee cave salamander. So we just kind of just finished a review, kind of reviewing overall cave obligate biodiversity in the United States and Canada. And so the current list of taxa is about 1,460 described species. About 930 of those are terrestrial, so we call those the triglobians. And then 530 are the aquatic species, so stygobionts, so stygo in reference to the river Styx in mythology. So that represents about 7 phyla, 19 classes, 56 orders, and so on. Of those triglobionts, the vast majority of that diversity is beetles. So almost 300 species across the nation, over 150 species of pseudoscorpions, so a type of arachnid that you see kind of pictured there is the guano pseudoscorpion, 140 millipedes, and over 120 spiders. And again, there's a number of undescribed species that have been documented in the literature, sitting in museum collections that are still awaiting description. With respect to those stygobionts, right? So that should be 530, not 430. That's a typo there. About 400 of those are crustaceans, just 10 fishes, and about 11 salamanders. But these fishes are where I've spent a lot of my career, working on. So my dissertation work was trying to better understand this group called the Ampliopsid cavefishes, which are quite diverse in the eastern U.S. and the Ozarks and the interior low plateau, including this guy, the southern cavefish, which historically has been considered one of the most widest ranging subterranean fishes in the world. It turns out it's a cryptic species complex. So what's a cryptic species? So you have a group of basically genetically distinct lineages that morphologically are either identical or very, very similar to each other. And so we're still in the process. Some of my colleagues, John Armbruster, Prasanth Chakrabarty, and some others, finally putting some names on some of these lineages that I had defined in my dissertation work over a decade ago. Salamanders, this is the Georgia blind salamander, Eurecia walsi. This is a species that's found in southwestern Georgia and adjacent panhandle in Florida. Look at the trogomorphy on this thing, right? So it's not purely albino. You can see and ready fours are still kind of there, but they're reduced in size and reduced in their expression. They have a really spatulate snout. You see those limbs kind of getting attenuate. They extend out from the body, right? That the reduced eyes that are regressing into the skull, itself. Really kind of amazing creatures that look really, really fragile. And so we've been working on a project here the last couple of years, kind of another round of surveys. We found several new populations, trying to get a better understanding of their genetics. It appears that there's kind of two major lineages that we're kind of finding that may have been isolated during parts of the Pliocene and maybe even into the Pleistocene, and have now come back into contact, maybe associated with changes in the sea level. So we're trying to kind of disentangle that. A number of crayfishes, and all these look very, very similar. They kind of differ with some subtle features with respect to their chelae and the rostrum and some of the of course the gonopods and whatnot. But again, just an amazing biodiversity. This is a cave amphipod. Looks big on your screen. It's only about 3 millimeters long, so it looks ferocious. A lot, again, a very diverse group that we find not just in caves and wells and springs, but they're found in shallow subterranean habitats outside of these karst areas. This is a new snail that we described a few years ago, the Tennessee cave snail. Again, looks big. It's only about 2 millimeters in diameter. Little bitty guy, just found outside Knoxville, Tennessee. Flatworms, a grossly understudied group, in general, but particularly underground. There's probably many undescribed species. They're purely white, right? Lack eye spots compared to maybe a flatworm that you may have dissected during, biology lab. Number of spiders, the subterranean sheetweb spider, it's our widest ranging species. And so most cave species tend to have really small restricted ranges. Some of these species are known from just a single cave system or just a couple isolated cave systems and a really restricted band of limestone or something like that. This guy's the exception. It's found in well over 700 caves in the Ozarks and most of the Interlittle Plateau and the Appalachian Valley and Ridge. And we just finished a genetic study, and they're all basically the same. So they're somehow moving around, maybe through the epikarst, or maybe there's other means of transportation. They've never been found on the surface. So they're kind of an enigma. They go against the grain on the typical pattern that we see with respect to the distributions of subterranean fauna. Pseudoscorpions, again, it's the southeastern cave pseudoscorpion, a relative to the guano pseudoscorpion, really common in cave systems associated with bats. Daddy longlegs, so cave harvestman here. You see reduction in pigments, right? More elongation of their limbs, and you see the palps there in the front. Number of millipedes. This is a really fragile ones. Peck's cave millipedes, Scoterpes, Sturt pecki, really sensitive to changes in actually humidity. So, you find cave systems as you go from the entrance into the deep cave zone, you tend to see more stability with respect to temperature, and you see basically changes in relative humidity where moisture in the air approaches, saturation. That's not always the case. Every cave is kind of different. If you've got cave systems that have multiple entrances at different elevations, you can get airflow, and in some cases, significant amounts of airflow during parts of the year that could be quite detrimental to this species. So we have a project with colleagues at NC State and Skylar Hopkins lab trying to better understand kind of cave microclimates and what impacts not only changes in temperature, but potentially changes in humidity and maybe even airflow might have with respect to the microhabitats for species like this. Springtails, look at the troglodyte on this thing. So, completely white, right? It's eyeless. But then look at those antennae. Basically, most of the length of that critter is just its antennae. Just extreme kind of troglodyte, kind of on display here. Here's a diploran, cave diploran, so a two-prong, bristle tail. Another kind of understudied group. Most of the diversity within this group is largely, they're kind of found in deep soil. But we do appear to have, some truly kind of troglodytic species as well. Beetles. So this is a cave obligate beetle. So they don't turn white like many other invertebrates do. They kind of get this kind of amber color that you see here, but they're eyeless. They're important predators in a lot of riparian cave systems, and again, a very diverse group, overall. So colleagues and I have been trying to better understand some of this diversity from a phylogenomic perspective recently. Another group of beetles, like cave fungus beetles, so instead of being active predators, these guys are kind of detritivores, right? They're feeding on kind of decaying organic matter. Where's this diversity found, in the U.S.? So, past cave biologists have kind of recognized 10 major cave or karst biogeographic areas across the U.S. So, what I've kind of shown here in this map, I've highlighted where those areas are at. Of course, you guys have one up in your neck of the woods, the Driftless area, where I'm at in northern Alabama is in that blue, the interior low plateau. So, not in the continent, and then Hawaii, which is all kind of volcanic karst there. There's a lot of gray that you see there represents kind of that karst at the surface that's not marked yet. So some of that, like in northern Indiana, is actually covered by glacial till. Other parts out west, though, so like the Mother Lode is an area that's been better researched, but there's a number of areas like in northern, parts of Arizona, Nevada that have significant diversity that was being uncovered here in the last decade plus. That 50 years from now, when we kind of revisit something like this, there's going to be probably many other, kind of biogeographic areas that are going to be kind of popping up, in general. It's just because there's been a lack of study, particularly in parts of the Western U.S. with respect to this. These are core diagrams that we kind of generated, kind of show based on those different 10 karst regions, how does this diversity kind of pan out by taxonomic groups there? And where's it kind of found at? So, for example, there on the left, we've got terrestrial biodiversity there. So the beetles, for the most part, you kind of see makes a big component. A lot of that diversity is found in the interior low plateau as well as the Appalachian Valley. In contrast, right there on the right, we've got the aquatic diversity. And so a lot of that diversity arises with respect to amphipods. What I want to kind of show there, though, is you might notice that there's this OTH abbreviation. You're like, what is that? That means basically other regions. So basically, other areas might be outside of a karst region entirely, or maybe not even associated with karst at all. So amphipods, isopods, and some of these flatworms are actually quite prominent in really shallow subterranean habitats that are outside basically limestone and carbonate areas. So in parts of the Great Lakes, right, in glacial till, northern Illinois, getting into Wisconsin, in Iowa, for example, basically drain tiles are a great area to be able to collect some of these species that are just found a few feet, maybe even below ground, but not associated with karst terrain. How's this diversity shake out by state? Um, so I've shown, again, we're talking Missouri, Illinois, just kind of gets a little bit closer in terms of the diversity for you all. This is kind of the terrestrial species. Unfortunately, Minnesota, where you're at, has about three that are described. I think it's a spider and a couple columbella, a couple springtails. With respect to that aquatic diversity, there hasn't been, to my knowledge, a described species of Stegobiont that's been reported. There probably should be a Stichobromus there somewhere, for certain. But given the terrain there, and you have a limited amount of karst, it's perhaps a little bit unsurprising. The greatest diversity is found in the areas where you maybe unsurprisingly have more caves and more karst. So parts of Texas, Tennessee, Alabama. You see Missouri stands out here, and Virginia stands out. We also have some sampling biases here too, right? There's been more cave biologists working in the eastern U.S. than parts of the West, or even parts of Texas. That's right. Texas is really kind of catching up, in terms of numbers over the last, couple decades. And then overall, this is where things kind of shake out, right? So Minnesota has three. Uh, this is a little bit older data set. Texas is, I think, north of like 255 now. Tennessee is around, 211. So we're still continuing to add species, slowly but surely. And again, this is just described biodiversity. This does not include species that are noted as being undescribed in the literature, but new. Now, with respect to their conservation status, I'm kind of showing here is these different taxonomic groups again. So we've kind of vertebrates on top. You see the salamanders and fishes, and then a variety of different invertebrates below that. Nature Reserve conservation ranks are on the left. IUCN conservation ranks are on the right. So if you're seeing red and orange and yellow, these are the kind of threatened statuses. So G1, G2, G3, they're on the left, and see the vast majority of species kind of fall into that. So it's looking at kind of proportion of species, overall. Again, most of these species that have been assessed, it's about not, I think it's about 80% of species of that 1,460 have been assessed by NatureServe. Unfortunately, a lot of those assessments are still fairly old. They're not within the last 5 years. In contrast, you look at IUCN, right, which is kind of the global standard, conservation assessment. Only 135 species, only about 9% of that 1,460 have been assessed. And you can see those clear biases, right? We're paying attention to the salamanders, we're paying attention to the fishes, paying attention to the decapods, the crayfishes and shrimps. Where a lot of this biodiversity is, though, things like, amphipods, things like beetles, millipedes, right? Um, Virtually none. And in fact, like beetles, there's only been, I think, like three beetles in their aquatic beetles of that 281 that species that we have here in the U.S. So it's pretty amazing, this kind of dichotomy that we're seeing. And this is a problem. This is something that is something that we need to address because it's going to impede our ability to effectively conserve and manage this biodiversity. Well, why are they threatened? Well, unsurprising probably to many of you, right? It's habitat change. So things that we are doing on the land surface have direct and indirect impacts below ground as well. So if we're modifying the landscape, we're changing the hydrology, we're changing the way energy is being inputted into some of these systems. We're introducing pollutants in general. So this is, Collins River in Middle Tennessee. This is after essentially water is flown underground and is issuing out of springs that are located, on the river itself. Anything that's basically flowing off the landscape, entering into these sinkholes, sinking streams, it's flowing underground, and the biodiversity there is being exposed to that. We see, gross, trash, particularly in rural areas. Folks don't want to take things to the landfill and have to pay money to get rid of their garbage. They still dump it to, they find an area in a remote area on someone else's property on the side of the road and just, dump it, and this washes underground. We see the runoff, right? Leading impacts. This is a sinkhole after a flood event, right? Look at all the the plastic and cans that's being deposited during these areas. So unfortunately, a lot of people still treat these areas as basically a natural landfill. Again, thinking that if it flows underground, it's not my problem anymore. Well, the reality is, someone is downstream who might be relying on that water source where it's sinking there and being exposed to these chemicals, and then using it, for their pets, for their own water. In some cave systems, unfortunately, we see evidence of this, right? So a lot of graffiti, a lot of trash. Of course, this with heavy recreational activity, you see risk of trampling for a lot of these sensitive cave invertebrates. I mean, most of the diversity, right, is really really small. It's less than half a centimeter in size. So if you're not paying attention to it, you're not going to see it. And we have sacrificial caves like this where, if they're not gated, every local knows about it. This is one in Middle Tennessee, kind of exemplifying this, in general. And this isn't from, cavers themselves. We call the people that do stuff like this spelunkers, right? These are the amateur cavers who maybe not even wearing a helmet or proper gear and clothing. And of course, they're just disrespectful to natural areas, in general. This is one that I found in a cave at Wheeler National Wildlife Refuge. So one of their popular caves by locals is supposed to be closed, but they don't have it gated and they don't have it well signed. It's constantly exposed to graffiti. This is actually a cave cricket that's still alive. So it was probably spray painted, the evening before we were doing a bio-inventory, that next morning. This was, really kind of eye-opening to me that this is sometimes the things that we're dealing with, and that mentality, that kind of disconnect with nature, and then appreciation and respect for nature, by some portions of our population. But if we're trying to summarize these threats, in general, so again, this is these same groups, right? And we've kind of broken it down by things like habitat loss there, habitat degradation, pollution, exploiting groundwater. What really stands out, though, is for a lot of those invertebrate groups, right? We don't have a good conservation assessment, to begin with. There's not any actual threats reported for them. We actually have very little direct evidence of threats. We have a lot of implied threats, right, in general. But if you look at the salamanders, the fishes there at the bottom, so crayfishes, much more work's been done on these groups. We know more about potential threats and impacts from said threats. But again, we have these kind of biases, in general. And of course, we have the same biases that exist in terms of actually protecting species. So with respect to like the Endangered Species Act, we have about 40 species that have been listed or proposed to be listed, with respect to subterranean biodiversity. And if you look at the percent of those, right, what stands out? We're in the bottom: amphibia, Nectiniopterygia, it's freshwater fishes, and amphibians, right? Despite the fact that they make a very, very small percentage of the overall subterranean biodiversity, in the U.S., the vast majority of those species, if they are severely threatened, have been listed or been proposed to be listed. And we see the same, right, there in C, right? It's kind of a different version, an older version, a book review, a book chapter that I did several years back, kind of showing those biases with respect to the IUCN Red List. Well, some of these other challenges that submarine biodiversity is facing really relates to some of our just general biodiversity, knowledge gaps. So, I'm not going to make you guys memorize this or have a quiz, thereafter, but just kind of showing and talking about some of these that you might be aware of. First, Linnaean shortfall. We're still finding these species. We don't have a good understanding of true levels of biodiversity, particularly in areas, out west. The Williston shortfall, that's with respect to the distributions of species. Our knowledge is largely incomplete, for many invertebrates in particular. And then we start to think about, other aspects of ecology, which would be important, right, for informing these conservation assessments and trying to prioritize species for conservation initiatives. understanding abundances, population sizes, life history, evolutionary relationships, right? Species interactions, all these types of things are largely unknown for the vast majority of species. We have a few, salamanders and fishes that we know much more about, maybe a few crayfishes, but again, most of those beetles, most of those amphipods, springtails, the flatworms, virtually nothing is known about much of their ecology, life history, and biology in general. And why is that? Well, part of it is, right, we can't easily study these habitats compared to life on the surface in many cases. So, for example, if we're going to study groundwater species, maybe we can access a cave, we could maybe sample from a well, we could sample on the surface at a spring where things get washed out, right, or a karst window. And that's useful, but this really is a three-dimensional matrix that's largely inaccessible to us as humans because we're just so much larger than a lot of this fauna and where it can go. Some other, challenges, a lot of the funding related to subterranean biodiversity, particularly for invertebrates, is fairly limited. Most of the funding in the last 20 plus years has been associated with bats, and rightfully so. White nose syndrome has been impacting bat populations since about 2006 here in North America. And we were, trying to get a better understanding of how it's being spread and maybe efforts we might be able to do to mitigate that. Unfortunately, there's really not been much that we have been able to do with respect to, helping to protect bat populations outside of, trying to do our part by decontaminating between trips if we're entering into cave systems. These taxonomic biases that I've already been mentioning, right? We see that with respect to researchers, largely focusing on vertebrates and then, the decapods, and then specifically cave obligate taxa. Virtually no one in the U.S. is working on mites or protozoans or copepods, in general. And so I say facetiously, who cares about them? Well, we should care about them, but we just don't have, the researchers to do so right now. The taxonomy crisis. We don't have, taxonomists or systematists who have that expertise to be able to identify a lot of these groups. So flatworms in particular, springtails. We don't have that many folks working on these groups. Diplurans, here in the U.S. Even, and so that limits our ability to, of course, describe new species, but to also determine, changes in range size, right? If there's range extensions or, species limits in general. And, really until the last, decade or so, we kind of had a lull in terms of overall number of really cave biologists that were working in North America. So kind of the heyday or the golden era of cave biology in the US is largely in the 60s, 70s, into the early 80s. Unfortunately, a lot of the um, researchers during that time period, they've either, passed away now or now retired. So, John Holsinger with amphipods, Tom Barr with beetles, just name a couple. And related to that, remember I mentioned there's 70,000, or more cave systems, in the US. Well, I'd estimate there's been maybe less than 3% of those have been even comprehensively bioinventoried, where we may have a good handle on the true level of diversity, in those systems. And then we've mentioned kind of the threats and conservation assessment, the lack thereof for some of these groups as well. And so, research in my lab over the last, really decade, or so has been trying to, better understand the distribution, the diversity, and try to fill in some of these life history gaps. It's a slow process, right? But some of the questions I would kind of ask, in my lab, or think about what are the true levels of biodiversity, right, in some of these systems, what sort of factors influence their distributions. We also take kind of a pop gen and phylogenetic approach to that. So for some of these groups, we're trying to better understand what are the major drivers of diversity, speciation. How do we see structure and genetic variation? What's that associated with? And so on. And then starting to think about, right, those human impacts and the conservation aspects, in general, and particularly, because we have, of course, limited funding and resources available. Are there certain areas, certain cave systems, certain species we need to be prioritizing for conservation and management that aren't already on the radar for state, federal, and local stakeholders? So just some highlights for some of this work. So over the last really 20 years, we've conducted, it's well over 1,000 biosurveys of nearly 700 cave systems throughout much of the U.S. And we're, continually growing, this information. So over 40,000 unique observations and documenting over 800 different species. At least 60 of those appear to be undescribed species that are awaiting description. The same token, my lab's been slowly kind of compiling literature and museum records across the US to try to develop essentially a continental data set that we can start to examine spatial patterns of diversity and look at those factors that might be influencing their distribution. And we're in the development of basically a data portal, with respect to this that can be used by researchers, but also the general public to kind of highlight biodiversity and make it a centralized area to find basically data and information about these species and about this biodiversity, quickly and efficiently. So some of the highlights that my lab has been able to uncover. The first species I was actually fortunate enough to be able to describe was actually the Hoosier cavefish. So this is an amblyopsid that historically has been treated as a single species. The northern cavefish, which ranged from basically, you see the map there, central parts of southern Indiana down to the Mammoth Cave area. Well, part of my dissertation was kind of looking at do we see any evidence of like genetic structuring, in this species? And so, in particular, is the Ohio River a potential barrier, to dispersal, in this group? And lo and behold, we see divergence between north and south of the river with no evidence of gene flow between them. Prasanta Chakrabarty at LSU and one of his students then I kind of assisted with this aspect, looked at a lot of museum specimens enough that we felt that it warranted recognition as a distinct species. So we named it after Indiana University, in part because Carl Eigenmann was an ichthyologist there at the university who first studied this group of cavefishes, and also because I'm a staunch Hoosier fan. Sorry, all you Minnesota fans in the audience. Well, this is the first time I ever went viral in terms of any of my research. So it started out pretty benign, just kind of documenting. Yeah, we found a new fish species. And then it kind of got a little bit weird. So the amblyopsids are a little bit unique as a group of fish because their neurogenital pore is migrated towards their throat. So some new terms were developed um, by by some reporters, like the neck anus. It's a little bit bizarre. And then it got really weird. Gawker got a hold of it, and that's when I stopped doing interviews after that point. Um, so be careful when you go viral with your research. We've been able to document some other, new species. I mentioned the Tennessee cave snail earlier, found in just three cave systems. It's a relative of another endemic found in Alabama. There's probably other snail, to be found and uncovered in additional populations or potential species. Again, it's just that, some of these species are so small and so specialized in their habitat that you really almost have to be there at the right place, the right time, and really looking for them to document them. This is a new silverfish, a cave silverfish that we found three cave systems here in Huntsville recently, and was able to describe that. It's related to species out in the Ozarks as well as Texas and Arizona. And there's many others that are still kind of in the works. We've been able to kind of document with additional survey work and rediscover several species of cave beetles and other fauna that maybe have not even been seen over the last 30, 40, in some cases even 70 years, which is encouraging, so suggesting that they're still around, and that includes finding new populations of some federally listed species like the Alabama cave shrimp, um, which is found basically around the Huntsville area. It's seen dramatic declines. There's only really a couple cave systems that are quite abundant, but it's encouraging, right, that we're finding new populations and rediscovering the Shelter Cape crayfish. So this is a single site endemic that hadn't been seen since the 1970s. So there, in that lower right-hand side, you're seeing basically a compilation of all the census surveys of Cape Crayfish. There's three Cape crayfish species that occur at this site. One being this was never in great abundance, but this single site endemic, and you can see a dramatic drop off there in the 1970s. That was associated with the construction of a cave gate that was not compatible with the grayback colony that was there. So they kind of vacated. That was an important source of energy. But also, this is in North Huntsville, a residential area. There's been a lot of tremendicide application that leaches in through the epikarst water and basically nuked the aquatic ecosystem there. So we saw the loss of several different species there that hadn't been seen in decades. Most recently, we actually rediscovered the Tennessee cave salamander there for the first time in 50 years. So maybe it's showing signs that the community is starting to recover slowly but surely. We've been trying to assemble these data sets and also try to get this out for others at the kind of the state level. In part, and this is work that we're still continuing to do. Again, to start to look at some of these spatial patterns of endemism. We work kind of hotspots that different state agencies, federal agencies might want to leverage if they're going to potentially try to purchase land. And we could generate, hotspots like this on a more regional scale. And again, the overall goal is to apply this at a really at a continental scale here, hopefully in the next, 5 years or so, as we're trying to finish up some of this data, compilation, in general. So we have aquatic richness there on the left, terrestrial richness there on the right, but you can see where these hotspots, kind of occur at, right? So southern Indiana, it really kind of stands out. You see the Mammoth Cave region there in Kentucky, my neck of the woods in northwestern, Alabama, and so on. Some other things that we're trying to do as well is, when I have students that are interested, particularly in groups like salamanders, is try to study their life history and ecology. So Kayla was a graduate student a few years ago, and she actually studied the reproductive ecology of a population of slimy salamanders that were reproducing in the cave system, here locally over the course of two breeding seasons. So she actually was able to, provide some important insights and show that, females aren't breeding every year in these systems. There's actually a high rate of egg loss. They're producing a few number of eggs, but over 50% of the nests completely fail. But the females are actually, in some cases, staying in the cave upwards of 9 months out of the year, which is a significant amount of time. And it looks like they might not necessarily be feeding, all that much as well. So she got some insights on that. So this is a female that you normally wouldn't expect this behavior. She's been probably sitting on a nest for a while. And, she's under illumination. She doesn't care about that. She notices that there's a little cave millipede there and gets a tasty meal. So Kayla's able to document, these salamanders feeding on things like cave beetles and whatnot, springtails, millipedes. I had another student, Megan Carter, kind of follow up on this to do some gut flushing analysis to really kind of see if they truly are feeding in the dark. And it looks like they're not. So this was probably a female that hasn't had a meal in probably several months and was just so hungry. Instinct took over. She saw the movement and was feeding there. Well, my lab's been kind of transitioning over the last few years to, try to supplement our traditional kind of approaches with using environmental DNA. So many of you are probably kind of familiar with this, by now. It's not necessarily as novel as it was, a decade ago. But, as you're sitting, listening to me as I'm sitting here, right, you scratch your face, you cough, whatnot. We're shedding, whether we want to admit it or not, DNA into the environment. Well, and organisms are doing the same thing in their environment as well, but we can potentially leverage that, from water, from soil, from ice cores, even from just swabs of flowers, looking at pollinators, spider webs, even just from the air now, to try to document, species and potentially monitor them. So, general workflow, for us is we might take water samples from a cave spring, a well, right, a cave stream, in general. We take that back to the lab and filter that, and then we extract DNA from that filter. And then predominantly, we've been doing these kind of single-based approaches. We designed an assay, right, to amplify DNA, usually mitochondrial DNA, a short fragment of that that's going to be specific for that species, and then be able to make inferences of whether it was we detect their DNA there or not, right? Well, we can also, because that same water sample might have DNA from many, many other organisms as well, we can also leverage that and we can take next generation sequencing approaches, right? And take basically a metabarcoding approach to then characterize entire communities. And that's something that, we're kind of moving towards, in general. Well, this can be useful for, of course, detecting species, particularly those in habitats like aquatic, groundwater habitats that are difficult for us to access. Help us to monitor, rare species, maybe detect invasive species. There's a number of different applications, for this that I'm sure many of you, are aware of. So we've been involved with the development of, some single species approaches for, some salamander species, cavefishes, crayfishes, other decapods, even amphipods. So the ones kind of highlighted here in orange that my lab has been involved with. And this is, there's been, several others, since then. I probably need to update this slide here. It's a little bit old. Our first demonstration of this was actually a federally endangered amphipod that's found in Washington, D.C. area. So if you've been to the National Zoo, you're basically right on top of the type locality for this little freshwater amphipod that co-occurs with a few other species there. So this is Dave Culver and Tanya Pepon actually sampling for the species. They occur in these basically wet spots in the wood. It's a hypotomenaraic seepage spring. And so the historic approach to kind of census them was to collect sort through the leaf litter here, collect individuals, and then you have to take them back to the lab and dissect out, certain body parts right, from another species. Well, that's kind of invasive and destructive, and it's for a listed species. You kind of don't want to be doing that. So we demonstrated we could actually detect DNA even from, aquatic habitats like this. Since then, we've been able to do this and apply this approach, to Cape crayfish here locally and several other species recently. Um, this is something that, the process of writing up now, but Kitty Dooley kind of extended this. So an endemic crayfish species that we thought was, more endangered, was only known from like 4 sites, versus a really closely related species that looks virtually identical, Cambarus jonesii, in purple there, which has a larger range. So that top map kind of shows what the previous range was assumed to be. Well, we got some real interesting kind of eDNA hits. We're applying these single species assays in multiple sites. And what we were able to kind of show then and go back to some of these sites and actually collect and genotype individuals from tissues we acquired is that the Speleocupi, the one that was known from 4 localities, it appears to actually be much more wide-ranging, still restricted. And then Cambarus jonesii, which we thought was in better shape and had a larger distribution, actually has a much smaller range. So we can potentially use eDNA to even help us delineate distributional boundaries for cryptic species complexes. We developed assays for some federally listed species. Unfortunately, we haven't had any, major range extensions with respect to that. But we can show that we can use this approach, including things like the Alabama cavefish, one of the rarest fishes that we have here in North America. And we're moving into metabarcoding now. So this is, something that, I've always wanted to kind of do, but again, try to better characterize these communities. And so this is something that over the last 2 years in particular, my lab has been collecting. We've collected over 300 from water samples from over 300 sites, including a project up in Alaska. So my grad student Jared Higgs just finished this up, comparing traditional biosurvey methods with eDNA. And so he sampled 13 cave systems, excuse me, 15 cave systems and 39 eDNA sites in general. We have another round from this past summer that we're going to be adding to this data set. So using traditional approaches, so things like visual encounter surveys and pitfall trapping, right, and doing Berlese extractions from leaf litter, we've documented at least 173 morpho species. Using eDNA, we've documented at least 360 taxa. There's a number of other essentially sequence reads, so ASVs in particular, that because of our limitation with respect to our reference sequence database, there's probably additional taxa that are there. So we see the significant difference between them. So again, kind of demonstrating that eDNA can be a very useful tool to help supplement these traditional approaches, particularly in habitats that are difficult to survey. We did see some taxonomic biases though. So eDNA did not necessarily do a great job of picking up things like springtails, diplurans, some of these other hexapod groups, probably in part because they're not necessarily well characterized in GenBank or the BOLD databases. And so they were more common in traditional approaches. And then some groups like some hydrozoans, surprisingly, some of the other aquatic taxa we're more likely to pick up through eDNA. Another question that we've been trying to look at is, how long does eDNA persist in groundwater? Well, on the surface, you get exposure to light, variable temperatures. eDNA, depending on the habitat, might last anywhere from hours to days to maybe a couple weeks. Well, with the stability of cave systems, basically occurring in complete darkness, right? You have the potential that it could last in a much longer time frame in general. But exactly how long is something that we're trying to consider. And of course, it has conservation implications. When we take a eDNA sample and we get a positive detection, we kind of want that to mean that that species was there, the day that we collected that sample, and not represent something that DNA that's floating through or been mobilized over years. And so I had a student, we're in the process of writing this up as well. We set up some mesocosms, kind of the entrance zone, the twilight zone, the deep cave zone at Shelter Cave here locally, and kind of tracked over time. We inoculated these mesocosms with lobster, something that shouldn't be in the environment. I ruined my wife's blender with respect to that, so I had to buy a new one for that. But what we're kind of showing here is that we see this trend that, yes, it's decreasing, but not reaching zero, right? A low concentration. We're still detecting it, in this case, after 300 days. Well, we extended this project. And this is data that Abby collected back in 2021. It's 2024, 2025. We were detecting DNA in the deep cave zone upwards of 4 years later. So again, these subterranean environments can potentially act as museums in some cases, for the DNA detections. So to kind of wrap things up, because I'm a little bit over in time here, kind of moving forward, one of the things that I think is critical for biodiversity conservation, particularly with respect to, habitats that are already kind of out of sight, out of mind, not necessarily on people's radar, is educational and awareness and outreach programs. So something that my wife and I have been very passionate about. And this is, this is my son. This is when he was 2 years old, kind of going caving, for the first time. Is to try to connect and get people excited at a young age about these habitats, even if they're not going to be a naturalist, not going to be a biologist, to at least have this exposure. Because as you guys are all aware, you guys, I'm showing my age. I was one of the last kids. I was born when I was an 80s kid, right? I'd be outside, all the time during the summer. I didn't have a screen in front of me. And so I still kind of have that connection with with nature, in general. A lot of kids these days, their only exposure is going to an aquarium, going to a zoo, something like that. And, if we're going to, conserve biodiversity and try to protect habitats, we need to have the general public behind us. But if they don't have that connection at all, it's really tough to do that. So this is something that, my wife and I have been passionate about trying to develop, and also instill just in our kids and in our our family in general, others that we always interact with, is just at least appreciation for nature. So this is River, with mom, when mom was pregnant with his little sister Evie, his first time finding a salamander underground. So again, and many of us who've been, kind of working in this field for a while, I kind of reached a point where I was getting really burnt out, right? You're constantly fed with negative news, right? The loss of more habitat, possible extinction here. And so it wasn't really until I had kids, and I was a little bit late, late in the game of having kids, that I kind of had this renewed effort to really promote biodiversity and conservation, in general. This is my daughter's, first Cape trip when she was about 9 months old. So with that, I'll shut up, and I really appreciate your guys' time. I just want to thank, over the last 20 plus years, there's been a number of different federal, state, and local partners who have assisted with funding, logistics, of course, private landowners in particular for allowing access to their cave systems. And this is the type of work, I'm highlighting it, right, but it takes an army to kind of do this. This is members of my lab that I want to thank over the last, several years who've assisted with these various projects, a number of important collaborators and in cavers, and many that I don't even have listed here. It really takes just a number of people, and I've always been really really grateful with respect to their participation. And with that, if we have some time, happy to answer any questions that you might have.
All right, thanks, Matt. I'm sure there are at least a few questions. Okay, Nick Phelps with a question over here. I want to ask if folks can, oh, he's clapping. If folks can unmute if they'd like to ask a question, unless for some reason their microphone's not working. Any questions for Matt? Are you able to see the chat, Matt, at all, or no?
I pulled up. Yeah, so you have a sense of how dispersal and gene flow works for cave fish. Are the caves connected? Yes and no. We have, yeah, some evidence in certain karst terrains. So particularly like around like Mammoth, for example, you have these horizontal bedded, really expansive limestones, right? And so using initially mitochondrial DNA to looking at rad-seq, to even looking at UCEs, in some of these areas we see distinct signatures that there's a dramatic amount of connectivity between cave systems in those areas, or at least the fish are able to maybe even access aquifers lower than the caves that we're able to access. In other areas, maybe not as much. And so, yeah, we can definitely make some inferences with respect to that. That's for some of the cavefish species. Now, for things like amphipods and isopods, they don't necessarily always show that same pattern. In a lot of the terrestrial species, they don't necessarily show those patterns. So, trying to understand still, particularly from a comparative framework, once we start to develop some of these data sets, are there some generalities? Do we see certain types of barriers being more important than others? Or do we see like a section with a subterranean, sheet web spider, something that we maybe would have predicted initially should have be a cryptic species complex, and maybe they're not moving around. They're showing the exact opposite, pattern to that. So that suggests that they're maybe utilizing another vector. Maybe it's, foresty. We've never been able to document that. Or maybe they're utilizing, the little cracks and crevices in the epicarps to get from one area, one cave system to another. And they're just much more mobile than some other species.
Anyone else? Trying to keep
Hey,
it.
man. Oh,
Go ahead, Nick.
Yeah, great presentation, Matt. I'm curious how you balance the trade-offs between collecting samples, like robust sample sets, with rare and endangered species.
Yes, that's a great question, and I've moved away from so full disclosure, I come from a vertebrate background, right, a hurt background first, where we're already kind of limited in that mindset and the number of samples we could take or try to take tissues, as opposed to maybe some traditional entomologists who might set out a pitfall trap and collect 2,000 springtails, right? I've always kind of had an issue with that. So a lot of our collections are using kind of visual. Um, encounter survey, type approaches in general, or for lazy extractions. And so, yeah, it's one of those things where I try to limit maybe no more than 10 individuals per morpho species at a given site, even for some of our invertebrates. And if it suggests that, oh, maybe we need to go back and do additional collections, well, we could potentially do that. Definitely, we no longer collect salamanders or crayfishes unless it's something that might be new or novel to science. We just get fin clips. Crayfishes the same way. A lot of these species appear to be fairly long-lived. We're going to have some evidence of that, and we could potentially have just through overcollection, there's some evidence this has happened in the past for certain populations. We could dramatically decimate a local population through overcollection. So the Shelter Cave, yes, it had those issues with respect to the loss of the bat colony, also groundwater pollution, but used to be a really large population of southern cavefish there. Well, it turns out around that same time period in the late 60s, early 70s, that some ichthyologists from Tulane University collected over 100 fish from that cave over the course of like 4 different survey events. That probably had a big effect, right, on that local population. So, I don't want to, have my name attached to something like that, that I, extirpated a unique species or something like that. So we try to be as ethical as we can in our collection. Next
That was a,
question.
yeah, that was a great talk. It's almost haunting that the DNA is hanging around for that long. Is it still helpful to have that information as a record of what you can still detect, even if it's not giving
Oh,
you?
certainly so. And we haven't, so in these, and full disclosure, right, with these are mesocosm. We were kind of simulating, right, if something died in like a rimstone pool, how long would it take to break down? So obviously, like a cave stream where you're getting water flow and probably a mechanical breakdown as well, that signature is going to be less. But I think it's important that we understand the ecology of eDNA in any system that we're working on, so we have an understanding of what it means when we do get a positive detection, whether it be in a metabarcoding analysis, right, or a single species detection and whatnot. So yeah, that's again, it's a useful tool. I definitely have viewed it as that, right? It shouldn't be a replacement for using some of our other approaches, but it definitely can be, something that we can potentially leverage. Or once we demonstrate that, hey, there's certain species that are here, or using it as a means to, okay, we're getting a detection in this watershed where we didn't know the species may exist. Maybe we need to prioritize some cave surveys right in this area.
Cool, thanks. I did my graduate work in Missouri, so seeing the karst topography was
Oh,
very
cool.
familiar. Thank you.
Yeah, I think is it Colleen? I think your hands up.
Hi, thank you. I was wondering if you have any thoughts on what helped to get into the eDNA work or some of the DNA work from more of a field background. I don't work in caves, but I'm also working to go from more of like a field ecologist type
Mhm.
structure to trying to add in some DNA work to help with our
Right.
research.
So yeah, my background is more evolutionary ecology, so I was already kind of interested in genetics. So like my master's work was like a phylogeography of Tennessee cave salamanders, looking at divergence with gene flow in that system, and then I was looking at cryptic diversity in general. So kind of that genetic aspect, ecological genetic aspect, was already kind of there for me. Um, and then it was just more of a we had just had an opportunity to have a small grant to see if we could try to apply this, and with the Hayes Spring Amphipod there in the Washington D.C. area. And that's really kind of how it kind of started. It was like, well, this is kind of interesting. Maybe we could apply this in other systems, other cave systems in general. And it's kind of just snowballed like many of the projects in my career. I didn't necessarily expect to be going down that path, but it just kind of did, so to speak. So sometimes you just if there's funding available, you sometimes have to chase the funding a little bit, particularly in this day and age, and at that time period, and even now, EDNA tends to be a fairly popular approach and topic. So there's a lot of folks doing using these approaches now in a variety of different systems. Mason?
Hey, great talk. My question is related to eDNA and metabarcoding. How do you
Mhm.
discern which identified like taxa are actually endemic to the cave versus like something that was terrestrial or external to the cave, I guess?
Right, right. And so if you're just taking like a single sample, right, it's going to be nearly impossible to do that from the eDNA alone. Obviously, if it's a cave obligate species that you're detecting and you have a reference sequence, you could probably make that inference, right? But how much of that is, water DNA that's passing through from the surface without having multiple samples throughout the system to kind of, and particularly at those point sources where they're coming in, you wouldn't be able to just at face value determine that. So, we have a couple studies, we haven't published this data yet, where we've actually looked at that various question, right? So how much are we seeing from the surface versus, truly actually occurring within the cave system, in general? That's a good, yeah, good question, a good point.
Hey, any last questions here? We're right at time. All right. Well, thanks a lot for a really interesting talk, Matt. If any wanted to hang around for a minute and talk to him, I'll
Yeah,
be happy.
happy to if there's any additional questions, yeah, or
I'll
just,
be hanging around,
talking
chatting
in
with Matt
general.
for a couple minutes here afterwards. So, thanks again, Matt.
Appreciate it.